Okay, and welcome to the first video from the Higher Human Biology course. Here area 1 of Unit 1, which is Division and Differentiation in Human Cells. I have a couple of slides to show you. I will talk you through the slides, and what I would like you to do, if you don't have these notes in your jotter, if perhaps you missed the class, then you take these notes. So it will require a wee bit of pausing and rewinding and double checking that you've got these notes down. For me, this is all the information that you'll require to allow you to do the past paper questions and the homework. So, to start off with, we look at two different kinds of cell that we find in the body. These are namely somatic cells and germline cells. Now, as you'll see, I have colour-coded both to show that they are different kinds of cell. Generally speaking, somatic cells are cells that we find in normal tissues in the body. Any cell that does not relate to reproduction. They make up tissues all around the body, so make sure you can provide a couple of examples of that, like skin tissue, nervous tissue, cells of the blood, for example, are also somatic cells. Remember that somatic cells are all diploid. Now you should remember from National 5 that means that the cell has two sets of chromosomes. Because this is human biology, we know that diploid number to be 46. And they only divide by mitosis. And again, you might remember mitosis from National 5 is where one cell divides into two identical daughter cells, both of which are diploid. Alright? So I'll give you a wee chance to look at that. In fact, you can pause it and make sure that you've got this note on somatic cells. Moving on to germline cells. The germline cells are any cell in the body which we relate to reproduction. Now that can include obviously gametes, which are sex cells, as we know from National 5 again. Gametes can obviously be sperm or eggs. They're defined as germline cells, but also the cells that divide to form those cells are also defined as germline cells. We call them germline stem cells. So germline stem cells are diploid. Got to be 46 written there, just taking you to that to mean diploid from here on in on this slide. Germline stem cells can do one of two things. They can undergo mitosis to provide more germline stem cells, or they can undergo a process called meiosis. Now this is a brand new process, and there is a wee bit of detail here, which I'm just looking at the screen, maybe not that easy to read, so make sure, listen carefully to what that says. Meiosis is involved, what is involved in meiosis is two stages. The first one, where homologous pairs of chromosomes line up. Now homologous pairs, just to give you a brief explanation, that will come up later in the course. Homologous chromosomes are because we inherit a set of chromosomes from our mum and a set of chromosomes from our dad. On the assumption our mum and dad are both human, they will both provide us with the same, effectively the same 23 chromosomes. Those two sets therefore have a pair of chromosomes which we call homologous pairs. So we have 23 from our mum, 23 from our dad, they come together to form pairs. Those pairs line up in the first, what we call the first meiotic division, and they are separated by spindle fibres, same way as we learned about mitosis in National 5. And in the second division, the chromatids that formed when the chromosome doubled up, they are separated. Now don't worry too much about the doubling up process, because we're going to talk about that in the context of DNA replication in the next topic. But just remember, meiosis consists of two stages. The first one, homologous chromosomes separate, and the second stage, chromatids are separated. And what results from those two divisions is 4 haploid gametes. And again, I've used the number 23 to denote haploid. That brings us on to the process of, we kind of dealt with division effectively, and now we're moving on to the idea of differentiation. Differentiation is a process where a stem cell, which as we know from National 5 are unspecialised, they don't have a particular role, they undergo a process of differentiation to become specialised and have a specific function in the body. That's how we've ended up with all the different kinds of cell that we have in our body. So I've just tried to highlight with a red line the most important aspects of understanding the definition of the word differentiation. Differentiation is where certain genes are expressed to produce proteins characteristic for a cell type. So to give you the background, a stem cell is unspecialised because it has no genes switched on or being expressed. The the process of differentiation involves choosing what genes are going to be expressed. Some will be, some won't be, and the result of some being expressed and some not being expressed is that some proteins are produced and others aren't. And the only difference between, for example, a nerve cell and a skin cell is that some genes in the skin cell are being expressed that are not being expressed in the nerve cell. So they contain the same information, but the information is being used in a different way. That's the process of differentiation. And if you are, and you will be, asked to define it, you're going to have to talk about genes being expressed and also the idea of proteins being produced. Okay, so a cell gets its particular role, gets its specific job in the body by certain genes being expressed to produce certain proteins that are characteristic for that cell type. Now, you do well to just remember that phrase, and when you're asked about differentiation, that's the definition you're going to give. Okay? It brings us on to stem cells. Stem cells, as we've just talked about, are undifferentiated, they are unspecialised. There are two different kinds of stem cell you need to talk about. We have embryonic stem cells, which are found in a developing embryo. What we know about them is that if you think about it, it makes sense that the cells in the embryo, they have to be able to differentiate into any cell type, because that's what we start off as. We all start off as a zygote which develops into an embryo. So those cells have to have the potential to become every kind of cell in the body, and they do, and that's what we call pluripotent. They are pluripotent, which means they can differentiate into any cell type in the body. Tissue stem cells, on the other hand, they are found in specific tissues in the body. So, you go to bone marrow, for example, is where you'll find the tissue stem cells for the blood. You'll find tissue stem cells in the skin. You'll find tissue stem cells in the digestive system. And different tissues have different tissue stem cells. They're involved in the growth, repair and renewal of cells from that tissue. Now, that is just lifted from the National 5 course, so you sort of should know that already. Growth, repair and renewal is the function of tissue stem cells within the tissue. And because they're part of a tissue, some decisions have already been made differentiation-wise. They don't have this same potential the embryonic stem cells have of becoming any kind of cell. They can only become cells from a particular type of tissue, and we call them multipotent. And we've said there, they can only differentiate into cells of the tissue in which they are found. And again, make sure that you pause this for a little second. I'll move out of the way and make sure that these are the notes you have on embryonic and tissue stem cells. Okay. Uses of stem cells. We do need to understand that there are two different kind of categories, if you like, of the uses of stem cells. The first one is they are used in research. Stem cells can be used as model cells. For example, if we want to test a drug or we study how the diseases develop. Now, it's worthwhile thinking about the ethical issues associated with that. We don't need to test in animals anymore. We don't need human subjects, because we can use stem cells, which are effectively models of how a drug or a disease might affect a cell as part of a living organism. So it avoids the use of animal testing or using human subjects. Stem cells can also be used therapeutically. So try and remember, when it comes to research, remember drug testing, remember studying how diseases develop, and remember the ethical issues. There's a lot of benefits for using stem cells in research. Therapeutic stem cells, or the use of stem cells therapeutically, can be used to repair damaged or diseased organs or tissues. And that's another one to try and just remember that phrase, right? Stem cells can be used to repair damaged or diseased organs or tissues. And give some examples. We have some of these in your locker. Skin grafts, corneal repair, bone marrow transplants are all good examples of stem cells being used therapeutically. That brings us back to the ethical issues. If, for example, we wanted to use an embryonic stem cell, as well as all the benefits that I just mentioned, we would also have to consider the fact that using an embryonic stem cell will require the destruction of a human embryo, and there are obvious ethical issues associated with that. Again, that's the note I'd like you to have on research and therapeutic uses of stem cells. And the final part is cancer. Now, cancer is in this topic because cancer is really just a fault in cell division, right? Cancer cells are just exactly the same as ordinary cells, except they divide uncontrollably. You'll notice I've underlined that because that's the phrase I want you to use in exam questions. They divide uncontrollably because they do not respond to regulatory signals. Cells will normally be told when to stop growing by cells around them. Those are regulatory signals. Cancer cells don't respond to them. They just keep on dividing uncontrollably. What forms then is a mass of these abnormal cells. It's called a tumour. You'll have heard of the word tumour before, but what a tumour is a mass of abnormal cells, all of them cancerous, forming because of this uncontrolled division. And then finally, if some cells fail to attach to the tumour and spread through the body, that forms what we call a secondary tumour, which arguably is more dangerous, because as soon as cancer starts to spread through the body, it's much, much more difficult to treat, much, much more difficult to remove, right? So very, very quick. I'll always try and buzz through this as quick as possible. I realise it's got to 10.5 minutes already, so you've got to take your time with this. All the notes that you need are on the board, so just go through the video, note down anything that you don't have, and that will give you the information that you need to equip you to answer the questions in the exam on division and differentiation.